Hi, my name is Pierre Addison from Stellenbosch University. This webinar will deal with the preservation of fruit flies obtained from routine trapping activities. These samples may contain a mixture of species, some of which may be rare, invasive, or of taxonomic interest. We therefore recommend that you keep all your samples at least until the end of a project or publication of the research. For long-term storage of material, all partners or collaborators should be consulted as to how long or where to keep the samples. The following points should be considered. Voucher samples are usually kept for first incursions of invasive species, new or unusual records, rare species, or material from new regions. Even common species should be kept as subsamples of voucher material. Voucher specimens should always be kept in a publicly accessible museum or other recognized institution. Plant research that would require a long series of material may benefit from series already obtained or those obtained from different time periods. Reference collection is material for which identification is confirmed by a relevant taxonomist. This can be used as reference material for research projects or training courses. Material for long-term storage should be kept in dedicated collections, such as museums, universities, or institutional collections. These may not be locally available, in which case the material can be transferred to institution with such a collection, preferably with a curator who is familiar with the group. For wet preservation, vials should be filled up with ethanol, propylene glycol, or formaldehyde. Propylene glycol is not flammable but quite sticky, so difficult to work with. Formaldehyde is toxic and destroys DNA. Vials should be filled as indicated above. This is to prevent flies from rotting. It is useful to have a pencil for making labels or pen that does not dissolve in alcohol, a fine paintbrush for moving specimens, or soft tweezers. For short-term storage, 75% ethanol stored at room temperature is sufficient. Ensure vials are closed tightly and check them regularly. For long-term storage and for genetic research, samples should be stored in absolute alcohol and kept at -20 to -80 degrees Celsius. This is to preserve the genetic information. Vials must always be properly labeled with the information as indicated above. You should also include the host if your samples were reared from a particular plant. It is best to place the label inside the vial as well, as the outside often fall off or smudge. Use a pencil or pen with insoluble ink for this purpose, preferably on Teslin paper. For dry preservation, samples can be kept in vials as dry specimens, as indicated above, or put between layers of cotton wool for protection, as dry specimens may become very brittle. They should, however, be completely dry, otherwise they can grow mold. This is not the best way of storage, and we recommend rather use them wet or pin. For pinning, stainless steel pins are best, either size 0 or use micro pins as in the image above. Pin the fly through the thorax as shown in the image in the middle. The pin should go through at an angle so that all features are visible on one or the other lateral side. Always apply a label to the pin. You can also add host and lure for more detail. Pinned specimens should be stored in airtight boxes, away from sunlight and high humidity, and preferably below 17 degrees Celsius, so keep them safe from pests. Also note that if you are pinning insects that emerged from laboratory-reared fruit, allow the adults to develop for a few days by feeding them with sugar and water before you pin them. As larvae are soft-bodied, they should always be preserved wet. Kill the live larvae by dipping in recently boiled water for a few seconds, then transfer them to a vial with 75% ethanol. Label as with dry and wet adult specimens. For more detail on preservation and dissection of larvae, please view our webinar on this topic. If you need to ship wet material, you will probably need to remove the liquid, as this can cause problems with the courier, particularly ethanol, which is flammable. Drain the liquid from the tubes, but add a piece of cotton wool with a few drops of ethanol to the vial. This will ensure that the atmosphere in the vial preserves the contents sufficiently during shipping. Ensure that the vials are not damaged during shipping. Dry materials such as pinned adults should be placed in small sturdy carton boxes with a soft foam bottom which keeps the pins in place. The specimen should not move around, so secure them in place as shown in the center image. Place the box in a larger box with padding to prevent further damage. You will need to follow local regulations for shipment of biological specimens, include keeping the relevant permits. We also recommend keeping a record such as a loan sheet. An example is given above. Thank you very much for watching our video on preservation of fruit fly specimens.